Welcome to the unit on bacterial metabolism. Throughout this unit, we're going to look at the various metabolic processes that are found in bacterial cells and how you can classify those organisms based on those metabolic properties. We're mainly going to focus on the catabolic properties, so the ways that organisms are able to gain energy. We're not going to talk in as much detail about the anabolic processes that are happening in cells. In this first video, we'll take a look at chemoorganotrophs, and we'll talk about what that classification means in a minute. We're going to focus on the beginnings of glucose catabolism in these organisms. Let's take a look. Throughout this section, we're going to be focusing on the catabolic metabolism, which is the energy-generating pathways that are found in a cell, as well as the pathways involved in breaking down large molecules into their smaller catabolites. Many of the small molecules will then feed into similar pathways. For example, carbohydrates funnel into glucose catabolism, while fatty acids will enter that same pathway, but at the acetyl-CoA step. It's important to remember that bacteria are very metabolically diverse, so that some species will be able to break down multiple different carbohydrates in different ways, while others might use lipids and others might not be able to break those things down at all. The type of metabolism that an organism can do and complete is really based on the presence of the enzymes that are part of those metabolic pathways. If the organisms have the enzymes, they can break those molecules down. If they don't have the enzymes, then they won't be able to use those as energy sources. Throughout this unit, we're going to discuss all of the various catabolic classifications of microbes that are listed in this table. However, we will spend most of our time discussing the two types of organotrophy, aerobic organotrophy and anaerobic organotrophy. These are representative of the majority of organisms that live within the human body, including both pathogens and the normal microbiota. Before we get to the details of the chemiorganotrophic pathways, I want to remind everyone that all catabolic pathways, no matter the classification, involve a series of oxidative reductive reactions. These redox reactions occur simultaneously, where electrons are stripped from one molecule and transferred to another. The molecule that has lost the electrons is said to be oxidized, but the molecule that has gained electrons is reduced. A common electron carrier in chemiorganic catabolism is NAD+, which acts as a shuttle bus in these redox reactions. An organic molecule is oxidized, losing electrons, which are picked up by NAD+, becoming reduced to NADH. NADH is then able to drop the electrons elsewhere, becoming oxidized back to NAD+. An easy way to remember what is being oxidized and what is being reduced is to remember the saying that Leo the lion goes grr. Leo for lose electrons oxidize, grr for gain electrons reduced. Also, if you're looking at the chemical structure, often when organic molecules transfer electrons, a proton comes along for the ride because its positive charge is attracted to the electron's negative charge. An electron and a proton together make up a hydrogen atom. So often when a molecule in biology has gained an electron, we will see the reduction in the form of an added hydrogen, which we can observe here with NAD+ here forming NADH and the addition of that hydrogen. Although we mentioned that various macromolecules will feed into chemiorganotrophic catabolic pathways in various locations, the one we're going to focus on is glucose, because that's the main energy source for many chemoorganotrophs, similar to yourself. In past classes, you have likely learned of glycolysis, the metabolic pathway that breaks one molecule of glucose down into two molecules of pyruvate. Glycolysis is also called the EM or the EMP pathway because it's not the only glucose metabolizing pathway in bacterial cells. You do not need to know every step of glycolysis for this class because it's not biochemistry. However, I do expect you to know what substrates enter the pathway and what products are produced. Besides the 2-pyruvate, glycolysis also produces a net yield of 2 ATP. This net yield comes because although the pathway produces 4 ATP, 2 here on this side and 2 on this side, it had to spend 2 ATP at the beginning to get the process started. Since it costs 2 ATP, you actually cancel out 2 of the ATP that were produced, which have just taken the place of the 2 that you spent. This means that glycolysis produces 2 new ATP molecules, which is the net yield. Glycolysis is also going to produce 2 of those electron carriers that we discussed, NADH, one here and one there. Many chemoorganotrophs have additional pathways that can break glucose down depending on what the cell needs to accomplish. The ED for Enterodorendorf pathway also breaks glucose or other sugar acids down to 2 pyruvate molecules. However, it only produces 1 ATP instead of the 2 that were produced during glycolysis. The other difference is that as opposed to producing 2 NADH, it produces 1 molecule of NADH and 1 of NADPH. The NADPH is typically used in redox reactions of anabolic pathways, building molecules up instead of breaking them down. This means that the ED pathway is beneficial to the cell when it needs a little bit of energy, but also needs to run some anabolic reactions to build molecules. The third glucose utilization pathway is the pentose phosphate pathway, which is the third panel in the image. The purpose of this pathway is in biosynthesis, or anabolic processes, instead of energy generation. So it is able to produce 2 molecules of NADPH instead of any NADH. This pathway is used when glucose needs to be broken down to use its carbon skeleton to build other macromolecules instead of generating energy for the cell. In the next video, we're going to start to continue with chemoorganotroph metabolism, starting from the pyruvate that we have now formed from glucose.